Chapter 16, Genetics. In this lecture, we're going to talk about how the different traits, genes, on our chromosomes are expressed. As usual, the objectives. You can either pause and come back here on this video, or you can go to the modules in Canvas and look at the PowerPoint without the audio. But make sure you know these different objectives. What is the basis of inheritance? So before understanding what DNA was, scientists figured it had to do with something in the sperm and the egg. They knew that those two came together to form the new individual. So somewhere in those cells there was a transmitting of information to the offspring, but they didn't know what and they didn't know how. Early theories called it a blob. The blob of dad's information blended with the blob of mother's information would come together. But what was that blob? Was it proteins? Was it the DNA? Was it an unknown material? And the problem that the scientists had was the variation amongst the offspring from the parents. If you took two of the same thing, why could it sometimes be different? So if you took two parents with dark hair, why could you still possibly get a child with red hair or blonde hair? This variation didn't make sense to them. Well, genes, genetics, help explain and predict inheritance patterns. A gene is a segment of DNA that carries the instructions for a synthesizing protein. And we've learned this before. This is a good definition to know. A gene is that sequence on the DNA that is going to transcribe and then translate to a specific protein. Genetics, these genes, can help explain why these children below look so very different, all in the human species. Analyzing their genes can also help predict the appearance of offspring to be born. Genetics determines body function and health, even though most of the genes that we're going to talk about have to do with our outward appearance because it's easier to explain. This gene is important for eye color. This gene is important for hair color, skin color, things like that. Most of the genes that we have do not have anything to do with our outward appearance at all. Most of them are internal. They're enzymes, enzymes and other proteins that are essential for our lives on the inside of our cells, the inside of our bodies. And I'm going to use a lot of the time hair color, eye color, skin color, things like that as examples because it's easy. But a lot of the times these particular traits are not just one gene. There's a lot of genes that are coming into play to create your eye color and skin color. But it's just visually easier sometimes to use those examples. Studying genetics also allows scientists to breed superior crops and doctors to track genetic illnesses. So the next couple slides, we're going to go over some different terminology. Some we are already aware of. Some we're going to learn that go a step further in what we already know. So as we already know, genes are a particular sequence that will transcribe and translate to a particular protein. While genes, those sequences on the DNA, are units of information about specific traits. They are passed from parents to offspring. We saw in our meiosis lecture how the DNA of the parent cell, the germ cell, is split up into the gametes. So each gamete has a subset of the germ cell, the parent cell's DNA, and then when those come together with another gamete, you will create this new individual. And so that's how the actual DNA, the chromosomes, are passed to the parents, from the parents to the offspring. And now we'll learn about the traits that are actually on those chromosomes and how they will interact, how the traits of the mother versus the father's chromosome, or vice versa, will interact with one another. Each of these genes has a specific location on a chromosome. And so a particular gene, let's say off to the right here, we've got gene C and gene D. Well, on the homologous pair of chromosomes, you find gene C in the same location. You find gene D in the same location. As you might notice, this is a big C, this is a little c. So there are variations of a particular gene. That's called alleles. There are different alleles of one gene. They're slightly different from one another. But again, you'll find them on the same spot, location, loci of the chromosome. Diploid cells have a pair of genes for a particular trait. So this is the pair. So on one chromosome, on one homologous chromosome, you've got the one gene. On the other homologous chromosome, you've got the other gene that corresponds with one another. Each chromosome of the homologous pair will only have the gene show up once. So when we're saying that there's a pair of genes, one is on one chromosome, one is on the other. And this could be, chromosome 1. This could be chromosome 17. It's just kind of showing how the different traits of the different homologous chromosomes have the same traits as one another. They might have some variations that are the alleles. Chromosomes are packets of genetic information, as we know, this long sequence of DNA with genes throughout them. Recall that chromosomes are a piece of DNA containing many different genes. The genes on those chromosomes, the location that they're found, is called the locus. When you are talking about more than one location, it's the plural of locus is loci. So on the bottom here, when we're looking at the locus of gene A, it will be found here. When you look at the loci of A and B, it would be here and here type of thing. And notice again how the locus of A is in the same spot for these two homologous chromosomes. So A is found in the same locus of both of these chromosomes. When two haploid cells come together, so two gametes come together to fertilize one another, a diploid zygote with two sets of chromosomes is formed. And this is at the end of, ultimately the end of meiosis, but it doesn't have to be. But when those two gametes that are produced by meiosis come together, you will then have a diploid cell with two of each chromosome, this homologous pair of chromosomes. And here is a karyotype again of a human genome, 1 through 23, 1 through 22 autosomes, and then the one pair of sex chromosomes. And as you can see, this one is a male, X and Y. Each chromosome each chromosome is a member of a homologous pair of chromosomes. So chromosome 1 will be a homologous pair. There will be two of them. Chromosome 8, there will be a homologous pair of them, one coming from your mother, one coming from your father. So homologous 8. We don't know which one is coming from which side of the family when we look at a karyotype, but usually when they're drawn, they're kind of drawn with, a pink color, a blue color, so that we have an idea of, this one is coming from the mother, this one's coming from the father. But it's kind of arbitrary. We don't really know until we start doing some math to figure out where it might be coming from, as in math, looking at a pedigree, doing things like that, some Punnett squares, which we'll learn about both of those in this lecture. The human has the 22 autosome homologous pairs and then the 1 sex chromosome homologous pair. Diploid cells can have 2 alleles for each gene. They don't have to have 2, but they can have 2. So the members of a homologous pair have the same genes, but might have different versions of those genes called alleles. And so this homologous pair has genes A, B, and D. A is big A. Well, on this homologous pair, it is also a big A. When it comes to B, there is big B and then little b. So there are 2 different alleles of the same gene on these chromosomes. Little d, little d. So this is the same allele. Now, what exactly are alleles? So the idea of gene A is going to determine the hair color of an individual. Well, big A can be black hair. And then little a could be brown hair. Or big A could be brown hair, little a could be blonde hair. These alleles may be identical, as we see with A, or they may be different, as we see with B. So the different alleles of a gene, the gene is a sequence of DNA. So if you have different versions of that sequence, those different sequences are going to be different. So the alleles of a gene have different molecular form. They have a different sequence of nucleotides. Now, they're close enough together, their sequences are similar enough that they're both going to produce a gene that is going to function somewhat the same, in the same way. These new alleles arise from mutations. So every once in a while, a new allele of a gene can pop up through a mutation. Basically, that mutation, a point mutation, a framework mutation, a deletion, addition, something like that. That is going to change that particular gene. That particular change, that mutation, could be a good thing, and it might stay in the population for a while. Could be a bad thing. Could be a lethal situation if this gene or this mutation is significant enough, or it could just be neutral, and there's not that big of an issue there. These different alleles, when coming together, will be interacting in different ways. And so you have, out of the different alleles, you'll have a dominant allele. And this is most cases. We will learn of some other situations later in the lecture. But a dominant allele will have a trait that hides the other allele that it is paired with. Usually this is written as the capital letter. So big A, little a. big A would be the dominant, just because it's written that way, big A, little a. We have the recessive trait will be a trait that is masked or hidden by the dominant allele. So this will be your little a. Now, for whatever reason, this dominance and recessive could be for different reasons. One big reason that some alleles are dominant versus the recessive is because the recessive allele is non-functional. So going back to those mutations where you had this neutral kind of mutation that it's, not quite bad, not quite good, that could be a situation here where the mutation of an allele just becomes non-functional, and the dominant is there, and the dominant will come into play. So one example here could be the color of a flower. So big R, little r. Big R, the flower is going to be red. Little r, the flower is going to be white or a whitish green. And so when the presence of the gene of big R is there, this allele of this gene, it's going to be vibrantly red. There is a particular protein that is being created, and it is causing this flower to be red. Well, in this case, the recessive is basically the absence of that protein. And so it has a gene, but the gene's not functional. And so if there was no dominant, if it was both chromosomes had this recessive, then this flower might be this whitish, greenish color that's naturally there without the protein at all. And so a lot of the times this is the case where the recessive is recessive because it just doesn't produce a functional protein, in this case, the flower color. But it's not always like that. There are a lot of other situations that the dominant is the dominant for other reasons. So when looking at allele combinations, I just want to reiterate, particular genes will be found on homologous chromosomes, where in that pair of chromosomes, one chromosome will have the gene, the other will have the gene as well. So one gene is coming from your mother, one gene is coming from your father. And when they come together in your DNA, you will have two. Now, in this case, we're talking about big A, little a. So we're talking about gene A. The alleles are big A, little a. You could have a combination of these alleles. And you could be big A, or little a, and that's what we're talking about here. And when these alleles are the same, when this pair of alleles are the same, it is called homozygous. Homo meaning same. So they are homozygous for gene A. Now, if you had both big A and big A, then that is homozygous dominant. If you had both recessive A's, the little a, that is homozygous recessive. And these are important terms to remember because I might not give you information about if it's big A, little a, big A, but if I say that it's homozygous dominant, you know that it's going to be the capital letter, or homozygous recessive, little letter. Heterozygous is when both alleles are present and they are not the same, and that is, in this case, big A, little a. And so in homozygous dominant, big A, you know that the trait of big A is going to show. So if big A represented brown hair, little a represented blonde hair, in this case, big A, you only have one choice. It's one or the other, and you're going to have brown hair. Little a, you only have one choice. It's going to be blonde hair. Heterozygous is when you're going to actually have the dominant, the big A, the brown, is going to overtake, mask the little a. And so in this case, you will have brown hair. So this version you have brown, this version you have blonde. So homozygous dominant, you have brown. Homozygous recessive, you have blonde. And heterozygous, you have brown. And so what I was just talking about there are genotypes versus phenotypes. As you remember, the brown and blonde, there were two examples of how you could have brown, only one example of blonde. Those are your phenotypes, and the genotypes are the specific alleles of the individual. So just to keep going with the genotype, so in that past example, the genotypes were big A, that's a genotype. Little a, that's a genotype. Big A, little a, also a genotype. Or heterozygous, homozygous, these are genotypes. They're talking about the actual genes, the alleles themselves. Phenotype is the individual's observable trait. So blonde hair or brown hair. That is the phenotype. What is seen, what is observable. Now, they're not exactly the same when you're looking at an individual. A lot of the times you can tell what their phenotype is by looking at what's present, but you might not know what the genotype is because it could be more than one example. So with the phenotype, if an individual had brown hair, you wouldn't know if it was big A, or heterozygous, big A, little a. You would know that there's at least a big A there, but you wouldn't know until you kind of started doing some Punnett square type situations. It's not always possible to determine a person's genotype from a phenotype. Just because someone has brown hair doesn't mean you know that it's big A. It could be big A, little a. But if a person has the recessive phenotype, then you could tell that person's genotype is little a. So if that person had blonde hair, little a. So here is stepping away from human characteristics. We'll get into the pea pod or the pea plant. Talking about genotypes and phenotypes. So a genotype represents an individual's two alleles for the one gene. And don't forget that the phenotype is the appearance or what is observable. The genotype will create the phenotype, which is the physical appearance. In this case, we're talking about the color of peas. So over here on the left side, we have lists of genotypes. Homozygous dominant, big Y. Y is going to equal yellow. Little y is going to equal green. So homozygous recessive is going to be little y. As you can see, its phenotype is green. In this case, the gene that is creating a protein that is a yellow color is absent here, and green is its natural kind of state without any colored protein. Heterozygous is big Y, little y. In this case is yellow because the dominant is masking the recessive. And so homozygous dominant, individuals with two dominant alleles, in this case big Y, and the phenotype is yellow. Heterozygous is one dominant, one recessive. You also have the phenotype of yellow. And homozygous recessive individuals have two recessive alleles, little y, in this case green. So look at the genotype and phenotype here. If you saw yellow peas, you would not know exactly what its genotype is. It could be one or the other. But if someone told you that it was the genotype was heterozygous or whatever, you would know what phenotype it was because you could calculate that. Gregor Mendel is the father of genetics. He was an Austrian monk, not Australian. He was Austrian. And his life's work was dealing with the pea plant, looking at the genetics of the pea plant. Well, he was also, a mathematician, a physicist, a botanist. But he used the pea plant to discover lots of different things that was taking place. These inheritable traits and how they passed on from one to the other, from parents to offspring. Now, Mendel did not know about DNA. I don't know if he knew the material existed, but he did not know how it functioned and how it worked and its structure. But he did know how traits were passed. He would mate these pea plants and look at the statistics of the offspring, how often a certain character showed up versus another character. And he did math to figure these things out, and he found patterns. And then these patterns came up with Mendelian genetics, which is kind of what we're going to be doing, looking at Punnett squares and things like that. Mendel's idea, a plant inherits 2 units of information about a trait, one from each parent. So without knowing about chromosomes and genes and DNA, he was still able to realize that there was some kind of a unit coming from each parent, and one of those units will be masking the other unit, dominant versus recessive, and he calculated this, worked it out in his lab. So he used the pea plant, Pisum sativum, and this particular plant was very good for him. It was small, it was easy to grow, it reproduced quickly, so it was a good example of something that he could use. If he used an oak tree, he might not. It might take years before he sees some of these adult reproductive traits. But not a pea plant. He was able to use them and look at these generation after generation pretty quickly. Now another thing about these pea plants that is very important to realize. They were true breeds of pea plant. Now, what is a breed? It might be easy to think of the breed of dogs, because that's something that we know more about. Breeds of dogs are man-made, artificially made. Same with the breeds of pea plants. Most of the breeds of agricultural plants and animals are breeds, which basically means for centuries, humans were the ones that determined which of these animals would be breeding with one another. And so they would breed for particular traits. I want to have a breed of dog that's smaller. So you'll take the individuals that are small of a certain group of offspring, and you'll mate them together, and the chances of getting smaller animals, smaller dogs, is going to be more likely. And so you keep doing that. You think about generation after generation after generation. lots of generations, you create what is called a breed. A breed is pretty much a group of this species that is homozygous for a particular trait that you were selecting for. So if you wanted a certain plant to be purple, and you bred and you bred and you bred them together, only the purple ones together for years and years and years, you're basically breeding out the other allele. And so just the allele that you want will be present in that group. Now, if you take this breed and you breed it with, you mate it with another breed, obviously now you're going to mix and match all of these up. But just think, breeds, they're not homozygous for every single trait in their DNA, in their genome. They're only homozygous for the ones that human has been selecting for, whether it's the color of the flower or the size of the animal, things like this. Did he know? Did Mendel know that these were true breeds, or that these breeds were homologous? That I don't know. There's a lot that Mendel did, that there are these little questions of, he did things really perfectly, which is great, but usually science is not as perfect as this. And so, I'm not saying that he cooked the books in any kind of way, but he came up with some things that were pretty right on. And did he do this by luck? I'm not really sure. So then he would take these different breeds and he would experimentally test cross them. So test crosses. Basically, in order to figure out information about these alleles, which one is dominant, which one is recessive, you need to do these test crosses to figure out which ones are going to show up and which ones are not going to show up, and then you run the numbers. We're just going to do a quick, Punnett square and figure these things out. But this was his life's work, dealing with one plant, dealing with, I think, 7 traits. Most of them were colors or shapes of the peas and the pea pods and things like that. So test crossing. He crossed 2 plants, the purebreds, with different traits. So he crossed 2 plants with different traits. An example here, purple plant, white plant. Both of them were homozygous. Not knowing exactly which is dominant or recessive, but they were homozygous just the same, purebred. These purebreds are called the P generation, which is parental generation. You could say P, purebred generation, but it does stand for parental. The offspring of this P generation is called the F1 generation. When you mate the F1 generation with one another, those plants, pea plants are also able to be self-fertilized, but you could take, if you're dealing with animals, you could mate the individuals of the F1 generation together, and you would get the F2 generation. Now, why do you have to go from F1 to F2? Why couldn't you just figure things out in F1 generation? Well, we will talk about that. You have to get to this F2 generation to figure out which of these alleles are dominant or recessive. So, we are going to learn how to do a Punnett square. I do not have a pen and a whiteboard right now to show you, so hopefully this tool here on the slide will be just as good. So, possible gametes. So what we're going to be doing, a Punnett square, is basically you're going to be writing down the alleles of the parents on the side and top, and then you're going to be filling in the squares to figure out the possibility of the offspring. Now, possibility. This is probability, flipping a coin. So remember when we talked about gametes being all equal in their ability to fertilize. So this is a random. Which one of these squares is it going to be? Well, sometimes the probability is 75% to 25%, so it's more likely going to be one or the other statistically. And that's what this Punnett square is figuring out. So possible gametes are listed at the top, egg or sperm. It doesn't really matter, top or bottom, male or female. And then you write the traits of both parents. So in this case, we'll be talking about, this is the F1, where the P generations are mating to create this F1 generation. So they're purebreds. So that means one is homozygous recessive. And that's why I have up here, parent one is little a, little a. And this is where you're going to draw it or write it. Little a over top here, little a over top here. So you're going to split up their alleles. Parent 2, you'll write here, and also homozygous dominant in this case, big A, big A. So I colored it so that you know that little a represents the white flower. Big A represents the purple flower. And so that's what kind of this is supposed to represent here. Then you're going to fill in your square. And this will tell you the probability of the genotypes for the potential offspring that will be created from the fertilizing of these two parents. So what's going to happen is you will bring down, you'll fill in this cell with the column and row. So you'll bring down the A, you'll bring over the A, and you get A, big A, little a. Why didn't I write it little a, big A? It's just the way it's done. The dominant is always goes first. So if I wrote little a, big A, yes, I would understand the situation, but it's just not the way it's drawn. And so I will fill in the rest of these doing the same thing. You're bringing down to fill in this this square, this cell. You bring down the little a, you bring over the big A. For this cell, you're bringing down the little a, you're bringing over the big A. And so they all get filled in, and as you notice, they're all the same genotype. And so this is our F1 generation. And as you might remember from the last slide, I said we have to go to F2 because F1 is not good enough. And this is the reason why. If you mate two purebreds, you're going to get all this dominant offspring. They're all purple because the big A is going to mask the little a. Now, I guess I did mess up a little bit. I said you have to go to F2 generation to figure out which one is dominant recessive. Obviously, we just did that. So you have to go to the F2 generation to figure out probabilities of offspring and to learn a little bit more. Mendel did more than just figuring out what was dominant and recessive. He looked at these percentages, these probabilities. So the frequency of offspring expressed as a probability. So if I asked you to mate these two, this homozygous recessive with a homozygous dominant, and then I asked you a question of how many of that offspring is going to be purple, then you would say this generation 100% will be purple. If I were to say what percentage, if I were to ask what the genotypes were going to be, the probability of the genotypes, you would say 100% big A, little a. So please do not get that confused. If I asked you the phenotype, you would tell me purple. If I asked you the genotype, you would tell me 100% big A, little a. You will get the question wrong if you answer it if I asked for genotype and you said it was purple. So please remember those two vocabulary terms. And this is a monohybrid cross because we are only dealing with one gene, which is A. Later, we'll do one of these Punnett squares dealing with two genes, so A and B type of thing. But this is a monohybrid cross. P generation coming up with the F1 generation just for gene A. So now we're going to mate the F1 generation with each other to figure out what the F2 generation is going to be. If you recall, the F1 generation was all big A, little a. All of the cells were filled in with big A, little a, so that is our only option. When I fill in the parents, I'm going to fill them in with these genotypes to figure out what the F2 generation is going to be. The F2 generation is going to be filling in this square. Now, don't be confused. I wrote parent 1 and parent 2. This is not the P generation. These are just the parents coming together to create the offspring. The parents are the F1 generation. So I could have wrote F1 parent 1, F1 parent 2, but honestly I forgot to do that. But please do not be confused by them being labeled parent. This is not the P generation. So we are going to fill in this Punnett square just like we did before with the parent genotypes. So big A will come down, big A will come over. We will do the next, little a will come down, big A will come over. The next, big A will come down, little a will come over. And the last, little a comes down, little a comes over. Now this is the possible offspring that can be born between these two parents, the F1 generations. So these are the genotype possibilities of the F2 generation. I said genotype because we're looking at their letters, their alleles here. When we look at the phenotype, you can see that 3 of the 4 are going to be purple. So 75% of the offspring is going to be purple because big A, that's the only option you have is purple. And these two are heterozygous, and the big A is going to mask the little a. So the big A is purple, and it's going to mask. So you're going to have all 3 of these, 75%, be purple, whereas 25% is going to be little a, or 25% is going to be white. And so this phenotype will be 25% white. Now, you could write it 75% purple, or you could put a 3-to-1 ratio. If you wanted to write it that way, I'm cool with both. And so the F2 generation is what we're looking at in the squares. They have a 1-to-2-to-1 ratio. I hope this doesn't confuse you. So this is how you write ratios: 1-to-2-to-1 or 3-to-1. You'll put the colon in the middle. So let's look at the phenotype first. The phenotype is what they look like. It is 3-to-1 because there are 3 of one phenotype and 1 of the other phenotypes. 3 purples, 1 white. The genotype, there's actually 3 different types of genotypes that are there. There is the homozygous dominant, homozygous recessive, and heterozygous. And so that is why it's written as a 1 to 2 to 1. Writing these ratios like this is pretty common. 1 to 2 to 1 for the genotype situation, and then 3 to 1 phenotype. If I asked you, give me the possibility of a homozygous recessive, how many of the offspring is going to be homozygous recessive, you could tell me 25%. You could give me the 1 to 2 to 1, but you would have to tell me which one of these is the homozygous recessive. I like percentages better when giving it, but I will use both of these, the percentage and the ratio, when I'm giving tests and things like this. But I will try to not make it difficult. I'm not trying to complicate things even more. I'm just trying to show you that that's how it's done both ways. So the dominant phenotype here is arising in 3 different times. The recessive is only coming up once. So phenotype, this 3 to 1, that's how the 3 to 1 comes. The 3 purple to 1 white. And so this is our F2 generation that has been created. And so when Mendel would look at the F1 and F2, he had these different numbers to look at, and he could calculate what was going on. He also went to F3 generation, F4, F5. He kept going to figure things out. And with his math, he was able to figure out about these different alleles and how they were masking one another. It might seem very intuitive and easy, but he was the first one in the world to do this, and he bred these plants thousands of times with one another. And so this is kind of showing the parent. So up at the top is the parent generation, and so you've got the two breeds here, the purple breed, well, it's not white here, but the white breed. And so these are homozygous. One is dominant, one is recessive, because they are breeds. And so when you mix the two, when you breed the two, mate the two P generations, you'll get this F1 generation. 100% are heterozygous. And so we're showing both here. These two are mated, these two are mated, but then we're showing that 100% of the F1 generation is heterozygous. And then I guess instead of showing that this one is mating with itself, we have this one mating with this one, and you split up the alleles, and the F2 generation is this whole 1 to 2 to 1 when looking at the genotypes. 1 big D big D, 2 of them are big D little d, and 1 of them is little d little d. And then the colors is this 3 to 1. So 3 of them are the purple phenotype, and 1 of them is the white phenotype, even though it's colored orange here. So Mendel observed the F1 generation only showed 1 of 2 of the phenotypes. In this case, they were all purple. That was the dominant trait. The recessive trait was the trait that was hidden, the one that didn't show up. The F2 generation had a 3 to 1 phenotype ratio of purple to white. And so this was what he was observing. It wasn't until later that he was able to calculate that the phenotype ratio was 1 to 2 to 1. Because this phenotype, or I'm sorry, this genotype, I'm talking about the genotype ratio, this genotype is not observed. He had to actually calculate this. It wasn't until later that he was realizing the different maths and that there are two alleles from these different parents and how they work together. But this we do know now. So yeah, Mendel studied lots of different pea traits. He studied, I think, 7 pea traits, but he mated these peas lots of different times. And so you can see, when looking at the color of the peas, he mated them. He had 8,000 offspring to look at. And these ratios aren't perfect. This 3-to-1 ratio, you flip a coin 100 times, you're not going to get 50 heads and 50 tails. But if you do it enough, you're going to see that statistically it's a 50-50 ratio here. But nothing is ever perfect. And so when he looked at the pea pods, the color of the pea pods, it came close to a 3-to-1 ratio. The tallness of the plant versus short was a 3-to-1 ratio. So I've got 2 practice Punnett squares to go over with you, just to make sure you know how to do these simple ones. So in this case, in poultry, chickens, the rose comb, which is a multi-lobe shaped comb, is controlled by the dominant allele, big R. Its recessive allele is little r, and it controls just a single comb. So I want you to give me the genotype and phenotype of the offspring produced from crossing a homozygous dominant with a homozygous recessive. So in order to do the parents, parent 1 is homozygous dominant, big R, big R. Instead of doing an oddly shaped circle, I colored it red. Parent 2, little r, because it is homozygous recessive. I colored them white, instead of red. And so now you have your parents, and so you need to fill in the Punnett square. So filling in the first square with big R, little r. The next squares are all big R, little r, bringing down the big R's and bringing over the little r's. And so that was big R, little r is 100% of the phenotype. I asked for the F1. The offspring, the phenotype, 100% comb, rose comb, the multi-lobe shape. 100% big R, little r is the genotype. So this is very similar to the parent generation creating the F1 generation. They're all big R, little r, which is your genotype, and then your phenotype is all rose combs. We have another practice Punnett square. So in seals, the gene for length of whisker has 2 alleles. The dominant allele, big W, codes for long whiskers. The recessive allele is little w, and that codes for short whiskers. So what percentage of offspring would you expect to have short whiskers if you crossed 2 long-whiskered seals? One that is homozygous dominant, and I'm going to write the parent one up here, big W, homozygous dominant, and the other parent is heterozygous, big W, little w. So for long whiskers, the big W I colored gray. So once we now have our parents, we're going to fill in our Punnett square. That top left would be big W, big W. Big W, big W. On the bottom, big W, little w, big W, little w. And then I'll color in gray for the ones that are long whiskers. And you might imagine it is all of them. They are all going to have long whiskers, which means your answer, or the question that's asking what percentage of the offspring do you expect to have short whiskers, your answer is 0%. And that's how this example is done. So Mendel's next question, does the inheritance of one factor, which is one trait, one gene, have influence on the inheritance of another factor or another trait? So does the seed shape have anything to do with the seed color as they're being inherited by the offspring? And so the way he did this, he used parent flowers that were homozygous dominant, so just as like before, and homozygous recessive for two different traits. And so in this case, the different breeds that he was using, one breed would have round and yellow peas. The other one would have oval and white peas. And so then he would keep track of both of those traits and mate them in an F1 generation and an F2 generation. This is called a dihybrid cross because we are looking at two different traits. The ones that we were doing before is called a monohybrid cross. We were only looking at one. Now this is as far as we're going to get, but you can look at trihybrid crosses, and that's 3 and 4 and 5, and you'll see how complicated this dihybrid cross gets. You can only imagine if you were to look at even more. And I guess depending on what you're doing, you may never need to do this, but I'm sure there are computer programs that can do it super quickly. So the dihybrid crosses track the inheritance of two genes at once. Two genes on different chromosomes can be combined into one large Punnett square. So spoiler alert, this whole the two genes being on two different chromosomes is very important here, but we'll talk about that later. And so I'm going to go over the creation of the F1 generation and the F2 generation. The F2 generation has another example on the next page that talks about the same things, just in a little more detail. So the F1 generation, your parents, you've got the two alleles, or the two genes, and homozygous recessive for both. Little r, little r, little y, little y. Over here, this is the homozygous dominant parent, and it is homozygous for both genes. Big R, little y, little y. Again, R is for the roundness of the seed, and the Y is for the yellow. So big R is round, little r is oval or misshapen. Big Y is yellow, little y is green. And so, just as you imagine, the F1 generation is done the same way as with the monohybrid crossing. The only gametes that you can come up with here is little r, little y, little r, little y. And so that's why it's drawn here this way, to make it simple. You could expand it out to here, but it would be little r, little y, little r, little y, little r, little y, little r, little y, because those are the only ones that you can do. So this one is drawn kind of simple because little r, little y are the only ways it can be done. Big R, big Y are the only ways it can be done. Now this is drawn as an egg and a sperm. And that's the only kind of little difference that's going on here. So the F1 generation, as you might assume, just like before, when you are mixing a homozygous dominant and a homozygous recessive, you're going to get 100% heterozygous. and it is 100% heterozygous for both genes. Big R, little r, big Y, big Y. So 100%, that is the genotype. 100% the phenotype is round and yellow. So then when you go to do your F2 generation, your parents are going to be heterozygous for both genes. Heterozygous for both genes. And this is a little more complicated when drawing this. So the parents can actually have these gametes, can be 4 different versions of these gametes can be created. And so you can have a gamete that's big R, big Y, because it's the mixture, big R, big Y, or big R, little y, which is here, or little r, big Y, which is here, little r, little y, which is here. So these are the only ways that you can combine those gametes to be created, because we're dealing with 2 traits instead of 1. And so if you're going to do this with one parent, you're going to do this with both parents, because they're both the same. They're both heterozygous for both genes. So when you draw this out, I didn't explain this earlier with the F1 generation, but when creating the F2 generation, you're bringing down both of these letters. You're bringing over both of these letters, and that's how you're going to fill in these boxes. So this box down here, you're bringing this little r, little y down, this little r, little y over, and that's how you get the 2 little r's and the 2 little y's. Now, your phenotypes are going to be written this way, 9 to 3 to 3 to 1, but I will show this a little bit better on the next picture. So a dihybrid cross, if I were to ask you to make one of these, which I won't, you will have to know, the outcome here, but the ability to do the monohybrid cross, you will have to do that on a test. But you will have to know how to understand how this is done and be able to read the results. So parent 1 is, this is F1 generation creating the F2 generation. So parent 1 is this heterozygote, big R, big Y, little y. And so this is, you separate the traits, the alleles this way. These are your possible gametes that this parent can create. You're going to do the same thing with parent 2, because it is also a heterozygous for these 2 traits. And so you fill it in the same way. And then just like the other paper, this is the exact same dihybrid cross, so don't get confused there. When we fill in, we're going to fill in the same way before. So you're bringing down your big R, big Y. You're bringing over your big R, big Y. So for each of these cells, you're bringing down and bringing over and creating what the new genotype is going to be. And so that's how we have all of these genotypes now. When we look at the phenotypes, and this is how it got that 9 to 3 to 3 to 1 ratio, there are 9 possible round yellow offspring, because R is round, big Y is yellow, and so all of these are going to create the phenotypes of round and yellow, and there are 9 of them. There are 3 round green. So it is, the phenotype is going to be this dominant round, but then the recessive green. There are going to be 3 of the recessive oval-shaped, oddly shaped seeds, and yellow in color. And then 1 that is homozygous recessive for both. So really, so this is how you get this 9 to 3 to 3 to 1 ratio of phenotypes. And so it's just kind of expanding on that 3 to 1, but a little bit more complex. I hope that makes sense. I would do another one of these, but it's very time consuming. So Mendel's theories, the theory of segregation. The two alleles of a gene separate when forming gametes. The gametes combine randomly in forming offspring. So this is the theory of segregation when it comes to two alleles. They will separate. Randomly when forming offspring, which is basically what we were saying when we were talking about meiosis. All of those gametes that are created in meiosis, they all have the same chance of fertilizing each other. Every sperm has the same chance of fertilizing the egg. Well, every chance compared to its chromosomes. The next theory, Mendel's theory, theory of independent assortment. Genes located on different chromosomes are inherited independently. The segregation of alleles for one gene does not influence the segregation of alleles on the other gene. So, if these genes are on different chromosomes, they will not affect one another. They will both randomly assort. They will randomly segregate, as we saw in that dihybrid cross. One will not influence the other. And if this doesn't make sense, it might make sense once we talk about what happens if they're on the same chromosome. And so again, these theories apply when genes are on separate chromosomes. The first theory is basically just talking about one gene. The second theory is talking about the two genes and how they're independently inherited when they are on separate chromosomes. So what happens if genes are on the same chromosome? They are linked. The term linked is if they are on the same chromosome. Punnett squares and the product rule assume that both genes are on different chromosomes and they will sort independently. When genes are linked, they are physically on the same chromosome. Therefore, they are not sorted independently. The linked genes are not inherited independently from each other either. So that pretty much means, if you're homozygous, if you're heterozygous, and every time one of those is going to be becoming a gamete, it's going to always bring the other gene that's on that same chromosome with it. So they wouldn't be separated independently. And so if we look at, in this case, we're looking at eyes and wings on flies here, doing a dihybrid cross. And so, as you might imagine, this is the parent generation, because it's homozygous recessive. Oh, and I guess this is not a parent. So this is homozygous recessive, and this one is heterozygous for both. And so if you were to do this Punnett square, this is what you would find if you did a normal Punnett square. These are the results. And so your phenotypes and your genotypes would be 1 to 1 to 1 to 1. They would all be found evenly across the board. But that's not what we find. We find that it's this 5 to 1 to 1 to 5. And it's like, well, why is that? Well, that's because they are linked on the same chromosome. And so they do not follow the rules of the Punnett square. If these genes, alleles, are found on the same chromosome, you cannot do a Punnett square to figure out the percentage of offspring. And again, that's because when the allele that's found on one chromosome is going to be moving automatically with the other gene that's on that chromosome. And so you cannot split them. Now, you could, for whatever reason, have crossing over that would take place, but that's extremely random. So genes on the same chromosome are linked. Punnett squares and the product rule here cannot be used if genes are linked, because inheriting one allele influences the likelihood of inheriting the other linked allele. So there are some other situations that don't follow the normal Mendelian rules. So when thinking of those theories that we saw before, those are the typical Mendelian rules, so Mendel's rules when it comes to inheritance. But there are some other examples that how these alleles can interact with one another. So we have something called complete dominance, which is this is what we've been looking at before. We have a dominant and a recessive. One of those is completely dominant to the other, and that's what we've been doing so far. Well, we also have this case of incomplete dominance, where the heterozygote, the one that's big A, little a, that one will have a phenotype that is somewhere in between the two homozygotes. So instead of the dominant being red, and if the dominant was red, the recessive was white, instead of the heterozygote being red as well, it's going to be pink. It's going to be somewhere in between this red and white. We have something called codominance, where non-identical alleles specify two phenotypes that are both expressed in heterozygotes. So that is a very complex answer there, but I'll try to break this down and we will definitely see an example. One of the big ones here is our blood type. And so you can have the blood type A and the blood type B. Both of them are codominant. So if you are blood type A, then that means both of your alleles, well, one of them, because you can also have O. So both of them are codominant. So if you have an allele that's A, then you would be blood type A. If you had just the B allele, then you would be blood type B. If you had both A and B, well, then you would be blood type A and B, or you would be AB blood type. We will show a slide in a couple slides that'll make that a lot more, hopefully make a lot more sense than what I just said. So incomplete dominance first. As I said, this is kind of this in-between heterozygote situation. And so this is a snapdragon flower. And so the big R is red. The little r is white. And so if you mix the two homozygotes together, you will get pink as your heterozygotes, not red. And so the F1 generation, if these were your parent generation, the F1 would be all pink. And so we'll do an example here. Okay. So then if we take the F1 generation and mix them together to see what the F2 generation is. So we have a pink flower, which is big R, little r, crossing it with another big R, little r. So our parents here. And I colored it red and white because big R is red, little r is white, even though our flowers are pink because of this incomplete dominance situation. So then we fill in our Punnett square. Big R, big R. Big R, little r. Big R, little r. little r, little r. So this is what our genotypes are for the F2 generation. Then if we look at what the phenotypes are, we see that anytime you have the big R, it's going to be red. Anytime you have little r, it will be white. But when you have the heterozygote, it will be pink, just like our parents. Well, the parents, which is the F1 generation parents. So phenotypes, white, pink, and red, and they are in the ratio of 1 to 2 to 1, just like our genotype ratio. In this case, the genotype ratio and the phenotype ratio are the same, 1 to 2 to 1. If I asked you which ones, what percentage of the offspring was going to be pink, you would say 50%. What percentage is going to be white? You would say 25%. Here I have another example of an incomplete dominance situation. So in radishes, we have two alleles that have an incomplete dominant relationship for shape. Big L is long radishes. Little l is round radishes. The heterozygote radishes are oval. So big L, little l will be oval in between the long and round. So we're going to draw a Punnett square that crosses a long with an oval radish. So parent 1 is long. It has to be big L because it's the only way to have a long radish. The other parent is a heterozygote because it's oval shaped. So big L, little l, and that will create an oval radish. So when we fill in the graph, or fill in the square, big L, big L, big L, big L, big L, little l, big L, little l. And then I drew the shape of the radish instead of dealing with color this time. And so the oval shape is not quite round and it's not quite long. It's this in between of the two for the heterozygotes. So the question is, what percentage of offspring will be round? So when you look here, we've got 50% will be long, 50% will be oval. So that means 0% will be round. Codominance. So we're looking at this ABO blood type. And so this is an example of a multiple allele system. So there are more than just big A, little a. We're dealing with more alleles than just two. The two alleles, big I superscript A and big I superscript B, or we'll just say AB, are codominant when paired. So when they are both found together, they will be both present, A, B. The third allele is little I, and this is the recessive. So if you had little I, that's when you have blood type O. Gene codes for the structure of the glycolipid on the blood cells. So the blood type that you are has to do with this glycolipid that's on the outside of your blood cell. That is what gives you your blood type. A or B or O or AB. And then the whole positive, negative, that's a different gene altogether. So there is another gene that's playing in here that'll be a positive or negative, so A negative, B negative, things like that. But we're just talking about the blood type, not the positive or negative. So these are the 4 types of blood type that you can be. Type A. In order to be type A, you either have to be 2 A's or 1A little I. To be type B, you have to be 2 B's or 1B little I. To be type AB, you have to have both an A and a B present. It's the only way. To be type O, you have to be little I. 2 recessives. That's the only way. And so hopefully this makes sense. Basically throwing in an additional allele here for a codominant situation. We're going to do a Punnett square for the blood type. A little differently on this one though. So a mother has type A. A father has type B blood. They have 2 children. One child is type O. What is the genotype of both parents? So you might be able to figure this out very easily in your head, but I'm going to use the Punnett square. But I'm going to use it backwards. So we don't know what the parents are currently, but we do know that one child is little I, because that's the only way that you can have a type O blood. Well, if this square is little I, then you know that the parents have to have an I as one of their alleles. It's the only way to get that little I. And so now that we know that, we can easily fill in the other allele for the parents because we know that the mother is type A and the father is type B. So this would be the mother, this would be the father. We know that this is the case because this is the only way to have type A blood if you have one I, little I. And so this is the genotype of both parents. Now, if you can figure that out in your head very easily, that's great. If not, and you needed to work that out, this is the way to do it. So this is the genotypes. A little I and B little I. When we fill in the rest of the square, so we know that this block down here is little I, but we're going to fill in the rest of these. Well, I guess the question first, what are the potential genotypes of the second child? Now we'll start filling these in. And so these are the potential genotypes of the child, the second child. The phenotypes would be AB, B, A, and O. And all would be 1 to 1 to 1 to 1 ratio, or 25% for each of them. The genotypes you would write out, big IA, big IB would be 1, big IA, little I, big IA, little I, little I, little I would be the genotypes. Sex-linked genes are unique. They have a unique inheritance pattern. So most of the different alleles that we were talking about had to do with chromosomes on the, genes on the autosomes, 1 through 22, the chromosomes 1 through 22. 23, chromosome 23, the sex chromosomes, slightly different because as you can see in the pictures, X is this large chromosome, Y is this small chromosome, so they do not have all of the same genes. The Y chromosome does not have all of the same genes as the X chromosome. So it is majorly different than all of the other homozygous, homologous chromosomes. In humans, females have 2 X chromosomes, X and X. Males have 1 X chromosome and 1 Y chromosome. So if you were to do a Punnett square, but instead putting an allele on the sides and top, you put the actual chromosome itself, it works. And so, X and X, X and Y, this is the male, this is the female. And then you fill in the Punnett square, X and X, X and X, so those are the female offspring, X and Y, X and Y. So you have a 50% chance of producing a female and 50% chance of producing a male. And then if you see, the only the gamete that is choosing or factoring in what sex it's going to be is going to be the male gamete. So for mammals, it is the male gamete that can produce the Y, and that will be the decision here. The male gamete will be the one that is factoring in if it's going to be a male or a female. The female is both X's, and she can only give an X. There's no other thing that she can give. So the male gamete will be the one determining the sex. And that's what I'm saying here. Which gamete determines the sex of the offspring? The egg will always carry an X chromosome. It's the only thing it can. It has two X's. The sex chromosome in the sperm, therefore, determines the offspring. If it's going to the sex of the offspring, because it has both X and Y that it can give at a 50% rate. So genes on the X chromosome are going to be a little different, especially if you are a male. So males only have one X chromosome. Whichever allele is on that X chromosome is going to be expressed because there is no other allele. There's just the one. So if it's dominant, it will be expressed. If it's recessive, it will be expressed because there's no other allele to go with it. Females have two X chromosomes. So they interact just like all of the other different alleles that we've been talking about. If it's it could be heterozygous, it could be homozygous, recessive, homozygous dominant. There are two alleles because there are two X chromosomes. So down below, we're looking at the way this particular trait is going to look out with males and females. So big R is dominant, little r is recessive. And so the way that they draw this here, the X is the sex chromosome, and then the superscript is the allele. So when you see this, you know that this is an allele found on the X chromosome on a sex chromosome. And so this female is heterozygous, big R, little r, and this male is big R, and then on the Y there is no allele. So when you see these two mating, this is the percentage of offspring. The females are going to be all showing the dominant phenotype. Half of the females will be homozygous dominant. Half of the females will be heterozygous. When it comes to the males, they will be 50% dominant and 50% recessive. So in this case, the only recessive that is going to be a phenotype will be the male. If you switched it up and you had the same female mating with a male that is recessive on the X instead of over here dominant, then you would get these offspring, or this potential offspring, which is the heterozygous female, homozygous recessive female, and then the males are the same, 50%. They are the same as over here, because the female is the same. Her gametes are the same. Big R, little r, big R, little r. Her gametes are going to be the one that determines the gamete possibility of the male offspring. The male offspring, the male is not bringing anything to the male offspring in this case. Just the female is determining this situation. So it's all the females, what her X chromosomes are. So because the male only has one X chromosome, males fall susceptible, much more susceptible to X-linked traits that cause problems. Color blindness is a recessive trait found on the X chromosome. And what you see to the right here, in this cluster of dots, there are different colors, and you should see, well, not should, if you see the number 74 written here, well, then you're not colorblind between these two colors. There are actually, I think, 3 different types of color blindness. This is a test for one of them. If you cannot see the number 74, maybe you have an issue with your screen, you're looking at it in black and white, or you may be colorblind. And so this hemophilia, color blindness is a recessive trait on the X chromosome. Hemophilia is also, it's a recessive trait. So this is a blood clotting disorder. Normal clotting in heterozygous females, homozygous dominant females, but then if you are a female that is homozygous recessive or a male with recessive, then you can have issues with blood clotting. We're going to perform a Punnett square with the X-linked trait of color blindness. So color blindness is an X-linked recessive trait. Big R, normal vision, and little r is color blindness. So in this situation, a man and a woman with normal vision have a colorblind son. What is the genotype of both of the parents? Again, you might be able to work this out really quickly in your head, but I'm going to do it on the Punnett square. So the mother, the father, we're trying to figure out their genotypes. Well, the father, we know, has a Y chromosome because he's a male. We know that he has an X big R because he has normal vision, and that's the only way that that can work. We know the mother at least has to have one X with a big R because she is also normal vision. Well, we have a colorblind son, so we know that this square has to be filled in X little r Y because there's a colorblind son, and so that means he's going to be down at the bottom. These are where the two boys will be, where the male offspring will be, and then up here will be filled in for the XXXX female offspring. And so if this bottom right square is filled in, then we know that we can fill in the second mother's allele because basically we're working our way backwards here, where the Y is coming from here, and then this X little r has to be coming from up here. And I had clicked the button already, and so the genotypes are the mother is big R little r, and the father is big R. There is no allele on the Y chromosome. Well, the next question is, what is the probability that they will have another child who is colorblind? In order to figure this out, we have to fill in the rest of this Punnett square. And so for the first, this first square, we put big R, both on the X's, because we bring one down, we bring one over. The next female is big R, little r. She's heterozygous, so she will not be colorblind. And then the other male will be big R, and he will not be colorblind. So only one box is filled in with a colorblind individual. And so the answer is 25% chance that the child will be colorblind. Now, if this question was answered differently and said, what is the possibility that their next son will be colorblind? Your answer would be 50%, because you only have two down here that are going to be males, and it is 50%, only one of the two. If the question said, what is the probability that their next daughter will be colorblind? Well, the answer is 0%, because there is no chance that they will have a colorblind daughter. And these are all possibilities of questions that could pop up. There's something called X inactivation that takes place in females. And this basically is preventing the double dosing of gene products. And so when it comes to the X's and Y's, when it comes to the X chromosome, it's a little different sometimes. All of these other chromosomes are meant to have two sets of alleles that are kind of interacting. But when it comes to the sex chromosome, sometimes that's a little different. And so some cells will actually inactivate a particular X chromosome in a female. This has to be a female because they're the only ones with two X's. They will inactivate a chromosome, and only one of those chromosomes will take place of what allele will show up. And this is quote-unquote random activation, inactivation. And not all animals do this, and not all types of cells do this. But when it comes to cats, their skin / hair follicle cells will do this. And so in these cat individuals, they will randomly inactivate one of the X chromosomes. Now, when this takes place, you will see this multicolor, example of the fur. And so, if one X chromosome has the allele for orange fur, and the other has an allele for black fur, color patterns can emerge when X chromosomes are randomly inactivated. So there are big patches of this random inactivation because this is happening during the creation of the embryo. And so when one cell will inactivate, and then it will go through mitosis, and then everyone after that will be inactivated as well. So that's why you will have these large patches. And so whenever you see fur like this, it has to be a female cat. It will never be a male cat unless that male happens to have trisomy, happens to have 3 sex chromosomes, which is XXY. In that case, inactivation can happen and you can have a male cat like this, but that is the only time. Just an interesting little tidbit. So now we're going to walk away from the Punnett squares, and we'll talk about the next tool that's used with genetics, and that's the pedigree. So this is an older kind of way of doing things, looking at like family trees, filling in some of the traits, a particular trait per pedigree, to kind of see some pattern that might be emerging, and seeing if something is heterozygous, dominant, or I should say if a trait is dominant or recessive and things like that. So a pedigree depicts family relationships and phenotypes. So pedigrees do not look at genotypes for the most part because they're not known. But if they are known, then you can have an example like down here where something is a carrier, and that would be you would know the genotype. So this particular pedigree is tracking achondroplasia, which is an autosomal dominant disorder, creating little people. And so this pedigree, like all pedigrees, the females are denoted by a circle. Anytime you see a circle up here, it is a female. Anytime you see a square, it is a male. And so these two have a line drawn together because they are a mating pair. Sometimes, too many times, you'll see that these two are married. This has nothing to do with marriage. This means that they are a mating pair. And in this case, we are looking at humans, but this doesn't have to be humans. This could be another animal, plants. This could be something totally different. And so you see the horizontal line showing them, connecting them together, meaning that they are mating. The line goes down, and then every line that comes off of this means that that is their child, that is their offspring. And so they have one offspring, they have two, they have 3, 4, 5 offspring. And it looks like 3 of those are female and 2 of those are male. Now, in this next generation, you see that this individual offspring has mated with somebody, with a female, and had children of their own. This female also has a mate, but no offspring. This individual has no mate or offspring, and this one has a mate and offspring. The coloring in of the shape means that the trait that this pedigree is showing is present. So when a shape is filled in, it means that that trait is present. If it's not filled in, then that means it is not present. Unless it's given to us, we do not know if this particular trait is recessive or dominant. And so there are certain patterns that you will see if the trait is dominant or recessive. Now, it was given to us that this is a trait that is dominant. So we know that this trait is dominant. And we'll see every generation has filled in shapes. And that is pretty common when it comes to dominant pedigrees. You will see that the trait is seen every generation. It's not 100%, but it's a pretty good indicator. Now, we do not have a carrier in here, and not all pedigrees have carriers filled in. Carriers mean that it is a recessive trait that we are talking about, and this individual is heterozygous, as in their phenotype is not showing the trait, but they have the allele for that trait. So that means if you see this, that it has to be a recessive trait that this pedigree is talking about. And it also, the different phenotypes of the individuals. But this is not always seen. Most likely, you'll just be looking at solids and filled in shapes. So in this pedigree, you will see that the first generation is not filled in. So that gives you a clue. if you're not seeing the phenotype in the first generation, but you are seeing it in the next generation, then that means it has to be recessive, because that's the only way that this would work. So you know that this is a recessive trait when you are skipping generations. And in this case, we've gone 2 generations before one pops out. 2 generations. Now, in this one, we see some slashes through, and that just means that the individual had deceased. It doesn't have anything to do with the trait, that this is a lethal trait or anything like that. It's just kind of, it's like a family tree showing you different characteristics. So this tool is used to follow inheritance of genetic traits, identifies individuals that have a specific trait. It could be genotype, but most of the time we're talking about phenotype, what is seen. In this case, if we were talking about a genotype, then you would see that one of these at least, both of these would have to be filled in as a carrier. So you can deduce the genotype sometimes, but not all the time. Usually these pedigrees are showing the phenotype. And down at the bottom, it says affected male, and that's just another way of saying that the male is showing that trait, and female. And this is an example of a dominant trait, another example of a dominant trait. Individuals with the condition are seen in every generation. They're affected. The ones that are filled in, again, are going to be homozygous dominant or heterozygous. The unaffected, in this case, will be all the little a's, the homozygous recessives. Here are some more examples. The one on the left, we're looking at recessive, and we know it's recessive because we have carriers. We wouldn't see that unless it was recessive. And this is showing the difference between autosomal, so autosomes, the 1 through 22 chromosomes, versus a pedigree that is showing an X-linked recessive disorder. And so in this case, for the X-linked, most of the time, a clue here would be that females are either all negative or only carriers, and the males are going to be the ones that you see are affected, or the positives. And usually this is a trait, something that is showing you that it is X-linked. Whereas this autosomal, down here you have a male and a female. Now, the fact that you have a male that is a carrier, if you're given carrier information, you know that it cannot be X-linked, because a male cannot be a carrier because he only has one X chromosome. So it's either positive or negative. There's no such thing as a carrier when it comes to X-linked. But this is not X-linked. This is showing autosomal. I hope that is informative. This is an example, the last example, I believe, which is recessive trait. And you're seeing, this is autosomal recessive, and it is skipping generations. You've got lots of generations that are being skipped here, with it showing up throughout.